Great. Thank you so much to the organizers and all involved bodies for this very special, great event. And also thanks to you for the chance to present some of my work here today. So as Guntram said, indeed, I want to tell you more about the more basic research we do on molecular mechanisms and how we try to connect them to what impact they may have on the global carbon cycling. And specifically, I'm going to talk about some work we do looking at bacteria killing each other with a question in mind how this can affect the biological carbon pump that Chris just mentioned already. But to start a bit more general, when I think about Earth and what keeps things up and running as we know them, I mostly think of microorganisms for the reason that they are really driving our biogeochemical cycles and provide a lot of ecosystem services to us. As an example, the biomass decomposition in soil is to 90% done by bacteria and fungi that convert all the biomass that accumulates back into minerals. Or when we think about food, what was already mentioned yesterday, food production, it's nitrogen-fixing bacteria that can fix nitrogen from the atmosphere and convert it into a form that's usable by plants, providing plant growth and productivity. But as Chris, I'm also baffled by the fact that microorganisms in the ocean actually carry out half of global carbon fixation, as mentioned, and this is what makes the ocean really a very crucial player at a time where we have too much carbon dioxide in the air and where we're thinking about long-term storage of carbon and carbon export that our ecosystems provide. So zooming in on this process, we have a lot of carbon dioxide, a lot of carbon in the form of carbon dioxide in the atmosphere. And simply by the difference in partial pressure, a lot of this dissolves in the ocean. And this purely physical process is well understood and often referred to as the solubility pump. But the process that can actually act on a short time scale and that can really contribute to a long-term storage of carbon is the so-called and already introduced biological carbon pump. And this refers to the fact that microorganisms mostly, or photosynthetic organisms in general, fix carbon dioxide in the surface oceans into biomass. And even though a lot of this biomass goes through the traditional food web and is quickly respired back into CO2, some of this biomass will sink into the deep ocean, and this is really the process that can contribute to export of carbon. And here to say, if we would not have the biological carbon pump acting all the time, it's estimated that today's CO2 levels in the atmosphere would be 50% higher. So this is a very crucial process. And the microorganisms aren't only playing a role in fixing the carbon, but also of this biomass that's sinking down, which is around 15% of carbon that was fixed at the surface. It's quite curious that only 0.3% actually reach the sea floor. And here it's again microorganisms that live in the ocean and that can find these particles and decompose them and solubilize them, which contributes to the carbon being retained in higher surface layers. So you can see a lot of this carbon from biomass can be, by these microorganisms, converted into anorganic or organic dissolved carbon in the ocean. And just to bring in another number here, this pool of dissolved organic carbon is nearly as great as the carbon that we have in the atmosphere. So I hope I could again show you that the microorganism and the processes they carry out are really essential to understand how much the ocean can be a sink for carbon. And there are some processes that are better understood, but a lot of uncertainty comes from the fact that we don't understand a lot of the processes that microbes carry out and how they interact with each other at the moment. So as an example, only over the last 20, 30 years, it became quite clear that viruses play a large role as they burst cells every day in the ocean, which frees around 3 gigatons of carbon every year. And this brought us a little bit to the question if also bacteria that we know from lab studies also very often possess killing mechanisms, if also this killing that happens between bacteria all the time, if this is yet another factor that affects microorganisms in the ocean and ultimately the biological carbon pump. So we set off to study the underlying processes here better, and we use lab experiments to do so. So thinking about just such a marine carbon source and bacteria that can degrade such a carbon source, displayed here as a purple bacterium, we wonder what happens now if there's not only this bacterium decomposing the material, but also bacteria that kill them around. And to study this phenomenon, we use microfluidic chips, which contain a main channel that supplies medium to cells that are seated in chambers that come off the main channel. And bacteria can grow here in a monolayer so that we can observe what every single cell is doing. And as you can probably guess, the blue cells cannot grow as they cannot decompose this carbon source here, but the purple cells decomposing the carbon source can happily grow and duplicate. And you can see that at the surface where blue cells and purple cells come together, cells get intoxicated and turned round and eventually burst and disappear from our imaging. And the curious discovery that we made here is that you can appreciate after the killing started, now the blue cells are actually starting to divide, even though they do not have a carbon source in the system that they can utilize. So our conclusion from this is that bacteria that kill their neighbors can actually recycle a part of the biomass from these killed neighbors and retain it in the community. And to understand these principles better, we would like to understand more quantitatively how efficiently this process happens. So we use this imaging data that we capture and segment and track every single cell. And after then identifying where lysis events happen, we look at numbers as how many cells need to be killed to create one new killing cell. Or we also look at the single cell growth rate of every single cell in the system and wonder if cells that are closer to lysis events, to bursting cell events, grow faster than cells that are further away from any from any cell-killing event. And lastly, we also use markers, so-called isotopes, to really track how much biomass from purple cells ends up in the blue cells. And these are all methods by which we try to quantify this phenomenon a bit better in a very simple setting. But trying to feed this back and hypothesizing what this could mean for the biological carbon pump, I wonder if killing by viruses, as we know, means that cells burst open, a lot of the liquid interior spills out as dissolved organic carbon. Some parts that are particular, like the cell wall, may continue to sink. But if this killing by bacteria means that actually a considerable part of this freed carbon is retained and recycled by the bacterial community. And we are very curious to understand how much this phenomenon happens worldwide and to zoom out a little bit. So one question we have is how common is this killing system that we're studying here a little bit anecdotally in the lab? And what we did is to look at data of marine bacteria. And what I'm showing you here is really in gray, the tree of life of bacteria. So at the tips, you see all the bacteria that we currently know from sequencing. And overlaid in colors, you see how often these bacteria were found in the ocean. Dark colors are organisms that were found more commonly. And we can now screen these organisms for if they have this killing system or not. And we find, displayed here by a color, that a lot of them contain a complete killing system or a subtype of it. And we think that this system is very common in organisms that we find in the global oceans. And we also now look at organisms that are closely related and quite similar in many ways, but where one has this killing system and the other does not. And we have the question in mind if we can actually see some adaptation of these organisms with the killing system on this biomass recycling lifestyle. So do they seem to be a bit less independent? Do they maybe not produce all the substances they need, but start to rely on gaining these nutrients from lysing bacteria? And lastly, we think it's very informative to not only look at who has this killing system, but how abundant are these killing bacteria. And here we look at an agglomeration of several expeditions, including the Tower Oceans data. And what we can already see is that there are indeed areas or depth where much fewer of the organisms have this killing system, less than 1%, but other areas where over 60% have this killing systems. And from these big differences, we would like to eventually get to driving factors that may contribute to bacteria with the killing system being more abundant in one place, but less in another, and maybe even trying to get an idea about how climate change might affect the abundance of this killing system. And to just finish by saying it's not only the oceans where you find killing bacteria, but also looking at bacteria from other ecosystems, we find that also there, a lot of the bacteria that were identified by sequencing contain these killing systems, and therefore we think there is value in looking more into their role on ecological functions that we're interested in. So to summarize, we think that bacteria that kill other cells around them can indeed recycle biomass from these killed cells. We found that these bacteria with killing systems are quite common in the ocean and also other ecosystems. And we find that the abundance of them varies strongly. So we hope that we can actually get at environmental factors that drive this, the benefit of these killing systems. And lastly, we wonder if it's maybe not only the viral killing that plays a big role for the carbon cycling in the ocean, but possibly also the killing between bacterial cells. And with that, I would like to thank my two lovely groups I have the pleasure to be hosted with, a lot of very active collaborators and advising collaborators, and thank you for your attention.
Thank you very much. Thank you.